Okay, hello and welcome. Today I'm going to be going over the 2024 AP Biology FRQ solutions. I'm going to go through all 6 of them in a couple different parts of this video. So we're going to start off with number 1, and just keep in mind that they haven't released the solutions or the marking criteria for this just yet. So these are what I personally think are the answers. They may accept a wider range of answers. We just have to wait till the marking scheme comes out. But I'm going to start with this first one. They did slightly change the format of the questions. They are not giving you the full question and then giving you like the part A, B, and C afterwards. So we're going to do this in sections and then go through. I'll give you as many possible options as I think that they'll accept. But again, there could be multiple answers for these. Just keep that in mind. Okay, so we're going to start off with number 1, which is one of the bigger questions. These are usually about 10 marks. Number 1 and number 2 should be about 10 marks. The other one should be probably around 4 to 6 marks each. So to start off with this first one, the start of the question says, crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the first cell division. So this first sentence is actually very important. It goes along with obviously this question that you can see here, but this kind of sets the stage for it, and this is going to be fairly important for the last part of the question. Now, A number 1 says, describe the function of the S phase in interphase. Now, this is a very straightforward question. There's no tricks in this. This is just general knowledge. So this is talking about your stages of the cell cycle when you're looking at not just meiosis, but the entire cell cycle. So I'll draw a diagram and I'll tell you guys what you should put. So this is the entire cell cycle. You have a sliver of it that is for meiosis, or what's known as the M phase. So this is the actual division phase here. So within the M phase, you have prophase, metaphase, anaphase, telophase. And then after that, you have your G1 phase, which is your first growth phase. And then you have the S phase here. And then you have the G2 phase here. Now, from here all the way around to here, so basically everything, not including mitosis or the M phase, is going to be interphase. So all of this is interphase. It's basically when the cell is not in its actual division process. So we're looking at this part right here, the S phase of interphase, and it's just asking for the general function. So I recommend you write full sentences, but just for time purposes, the function of the S phase is DNA replication. That is the only function of the S phase. Very straightforward. I would just write full sentence saying the function of the S phase in interphase is DNA replication. I always remind my students that S, I usually remember, stands for synthesis, which is the DNA synthesis. So obviously you're going this way. So before you go through meiosis, which is when you obviously split into multiple cells, you do need to first replicate your DNA so that you make sure each one of your cells has the proper amount of DNA, the full 46 chromosomes if you're human. So the S phase is where the division of the DNA happens, and then you get the division of the actual cell in the M phase. So this is the first one. Okay, so this is the next part of the question. I'll read through the whole thing and I'll highlight some important parts. So this one says, some regions of a chromosome called hotspots display a higher frequency of crossing over than other regions do. So this is something that we briefly go over, but they do give you the definition here. So we're looking at places where crossing over happens at high frequencies. So crossing over is suppressed in chromosomal regions near the centromeres. The centromere region of a duplicated chromosome includes a collection of proteins that form a structure called the kinetochore. You learn about all of this in Unit 4. Scientists hypothesize that one or more of these kinetochore proteins are responsible for suppressing crossing over around the centromere. So they believe that around the centromere, which they give you a diagram here, the crossing over should be suppressed. So there should be less crossing over near the centromere, likely just because there are like bulk proteins there. So to investigate their hypothesis, scientists modified chromosome 8 in yeast such that in one cell, one chromosome from the pair of homologous chromosome 8s contained the gene encoding red fluorescent protein, also known as RFP, while the other chromosome formed a pair containing the gene encoding green fluorescent protein, or GFP. the cells expressing RFP emit or also give off red light, and then the cells expressing GFP emit a green light. The models of the modified chromosome 8s before and after crossing over are shown in figure 1. So they do give you a figure in this one. Always read your figure captions. It says, models of modified chromosome 8 used in the experiment A before and B after crossing over occurs at the hot spots. So this is before crossing over. You can see that you have 2 homologous chromosomes here. Again, always remember your difference between homologous chromosomes and sister chromatid. These are homologous chromosomes. One of them has RFP, the red light, and then one of them has GFP, the green light. And then when they do crossing over, you guys know that the 2 inner sister chromatids are going to swap, and they're going to swap somewhere within this hot spot, right? Because we said it's going to be suppressed somewhere near here, but there is going to be a lot of crossing over in the hot spot. So after crossing over, there's likely a couple different combinations of what could happen here. However, like depending on how high up you're doing the crossing over, but they just give you one version here where you had crossing over of those 2 inner sister chromatids that are attached to 2 different homologous chromosomes. And again, homologous chromosomes are not identical. So you did have crossing over that caused this RFP chunk here to cross over and swap with the chunk that is here. And then you end up with this inner sister chromatid having nothing because it gave the RFP away during crossing over. And then this inner one here had the GFP and the RFP because it maintained it or gained it during that crossing over process. Okay, so this next part here is not actually about this diagram. It's a pretty general question. It asks you to explain why some haploid cells formed after meiosis in the experiment will only have one fluorescent marker. Okay, so they'll only have one fluorescent marker, meaning after crossing over when we're looking at this, they will only have either GFP or RFP. Now, it won't be all of them. As you can see, some of them might have 2, some of them might have none, but some of them only contain one of the markers. And the reason for that is if we're looking at meiosis, meiosis has 2 steps. So for meiosis, you have 2 divisions. The first one is going to be your division of homologous chromosomes. So this is after crossing over. So you do crossing over and then you separate your 2 homologous chromosomes. So one cell will contain this chromosome and one cell will contain this chromosome. And then after, you have meiosis 2, which separates your sister chromatid, which actually breaks the arms off like this so that you only get one strand of this chromosome. Now, that is the reason that they may only have one marker, because after meiosis, you're not ending up with a full duplicated chromosome. You're only ending up with half. So if we're going by this model here, you should end up with 4 haploid cells, as you usually do with meiosis. This one should contain, one arm of the chromosome with the RFP. So we'll put a little RFP here. And then one of them should contain this one that has no signal. The next one over here should contain one here that has 2 signals, the GFP and the RFP. So we'll put an RFP marker here and a GFP marker here. And then the final one, which is this last chromosome arm here, will go into its own gamete, and that will actually only have the GFP. Okay, so just due to how the chromosomes separate during meiosis, and it's really not that specific to this question, you just need to know that at the end of the day, when you finish meiosis, you're going to end up with 1 chromosome in each haploid. And since for the haploid cells, you can have one of these markers. So these 2 both have only 1 marker. And so you didn't have to draw all this or write all of this. Frankly, the answer for this one, what I would put, so it just says explain. So you only really need 1 sentence for this. Explain why some haploid cells formed after meiosis in this experiment will have only 1 fluorescent marker. So for this one, the reason for that is that you're going to have haploid only contains haploid 1 copy of each chromosome. And you can also say because there's 2 divisions, You can keep expanding on this. So overall, haploid cells, which are what you get after the end of meiosis, is 2 divisions, and then you're going to end up with only one unduplicated chromosome in each one of your gametes. That's why some of them only have one marker. They're not getting both of the duplicated chromosomes, they're only getting one, so one marker. Not for all of them, but some of them do, like they mentioned here. So it's just because there's 2 divisions and you're going to end up with one chromosome in each of your cells. Okay, so that is A number 2. And then moving on, you have another graph here. And in addition to the question, it says the scientists then investigated whether attaching individual kinetochore proteins to a specific DNA sequence present in a known crossing over hotspot on chromosome 8 affected the frequency of crossing over at this location. In their first experiment, they examined 3 groups of yeast cells containing the modified chromosome 8. Group 1 contained no kinetochore proteins attached to the hotspot. So right here, they're giving you the parameters of the experiment. So group 1, no kinetochore proteins. Group 2 contained kinetochore protein CTF attached to the hotspot, and group 3 contained kinetochore protein IML attached to the hotspot. You don't need to know anything about these proteins. They're just telling you that those are the conditions. So for each group, the scientists determined the frequency of crossing over between the RFP and GFP genes. To determine the frequency, the scientists added the number of cells emitting both red and green light to the number of cells that emitted no light and divided by the total number of cells. Now, this part probably confused a lot of students here, but frankly, it's not very important for the question. They're just telling you how they got the metric over here, which is the frequency of crossing over. But frankly, you can just take that at face value. You don't need to think about any of like the light emitting or anything like that. So if we look at our diagram here, again, always read your figure captions. It says the frequency of crossing over in a hotspot on yeast chromosome 8 for cell groups treated with different kinetochore proteins. Error bars represent plus or minus 2 standard deviations. Okay. So you have your x-axis, which is attached kinetochore protein. You have 3 groups, which we've already mentioned here. You've got your no kinetochore proteins, CTF kinetochore proteins, and then IML kinetochore proteins. And then on your y-axis here, you have frequency of crossing over as a percentage. Okay, so let's go into this. B part 1 is very straightforward. It says identify the control group for the scientists' first experiments shown in figure 2. So they're asking what is the control group for this. So this is a very straightforward question. You guys should remember that control groups are what you're comparing it to. Now in this experiment, you're trying to see the effect of having different kinetochore proteins attached to that hotspot. So your control group, what you're comparing it to, is going to be the one that has no kinetochore proteins. So your control group is actually going to be the one that has no proteins, because you're going to take your CTF, you're going to compare it to this, you're going to take your IML, and you're going to compare it back to this to see if there's a difference. So your control group, all you would have to say is the group with no kinetochore proteins. Okay, so that one is pretty straightforward again. If we go to part 2 of B, you have a follow-up experiment, which says, in a follow-up experiment, the scientists created a modified version of CTF in which the DNA binding portion has been removed. They compared the frequency of crossing over in yeast cells in the presence and absence of unmodified CTF with that in yeast cells in the presence and absence of modified CTF. The data isn't shown. In the follow-up experiment, justify why the scientists use a modified CTF protein that is unable to bind to DNA as a control. Okay, so they don't give you any data behind this. They're just asking you the reasoning why you would want to use a CTF protein that can bind and can't bind. Okay, and the reason for that one is it's also just going to be a control. So the whole point of these kinetochore proteins is that they physically bind to the DNA to affect crossing over. Okay, so if you have one that is actually physically unable of binding to the DNA, that's going to be a really good control. So they use the modified CTF protein that can't bind, and then they use the regular CTF protein that can bind. So therefore, you can see directly as a control of comparing both of them, whether DNA binding is something that's affecting the crossing over frequency. So for this one, you can do a couple different options that I've seen. Is it allows for a direct comparison because, again, it is going to be a control. So you're allowing a direct comparison because you're exposing both to CTF. However, one is not actually binding. So that's actually going to be a much better control than the no kinetochore proteins in terms of a direct comparison. So it allows you to directly compare and see if DNA binding is affected. DNA binding affects crossing over. There's probably a couple different ways in which you can say this, but overall, it's just used as a control. And the main point of controls is going to allow you to make a direct comparison and see, specifically in this example, if DNA binding affects the crossing over, or really how the CTF protein in general, because the one that's modified shouldn't bind in the first place, just seeing a better comparison of what it's actually doing. Okay. And then 3 is asking again the independent variable in the follow-up experiment. So the independent variable of the follow-up experiment. So make sure you're not doing the independent variable of this one over here. This is our follow-up experiment. So for this one, you're taking modified CTF protein without DNA binding and then regular CTF protein, and you're looking at frequency of crossing over. Your independent variable is always your x-axis, so the thing that you're changing. So your independent variable is whether or not the CTF is modified. You can say this in a couple different ways. So if the CTF is modified, you can say, if the CTF has the DNA binding portion or not, anything like that, but that's what would be on your x-axis for this one. Your y-axis, your dependent variable, is your frequency of crossing over. Okay, so that is going to be B. Now, if we move on to C, it says, based on figure 2, describe the effect on the frequency of crossing over when CTF is attached to chromosome 8 compared with the effect when IML is attached to the hotspot. So that takes us back to figure 2. You're basically just looking at the effect of CTF and IML. So we'll take a look at this. And again, like I said, it's really important to establish in this part right here which one is the control, because you're ultimately going to be comparing it back to this to see if it's increasing or decreasing. So for this one, you can see that CTF is decreasing because this is with no protein. And then when you attach CTF, there's a decrease in the frequency of crossing over. You can also mention that it's significant because if you look at the standard error bars, there's no overlapping. And for IML, there is no change. So they're pretty much even when you look at the levels of crossing over for no protein and IML. So there was no effect. So I don't know if this would be 1 mark or 2 marks if you needed to say both of them separately. So for CTF, it would have decreased crossing over. And again, write these in full sentences. And then for IML, there's no change. And then I would maybe mention that it's significant, just in case. Again, I don't have the marking scheme for these, so I put as much information as you can. However, you likely wouldn't get a mark off if you didn't mention that it was significant. I'm just putting that just in case. Okay, so now for part D, it says predict the effect of the number of copies of chromosome 8 likely to be present in the resulting daughter cells when CTF is attached to the hotspot. Okay, now this one is the rather difficult one, and it requires you to go all the way back to what I highlighted at the very beginning. So for D part 1, so we're looking at the number of copies of chromosome 8 likely to be present, resulting in daughter cells when CTF is attached. So we've already mentioned that CTF should decrease crossing over, okay? And part 2 is also providing reasoning, so I'm just going to answer these all as one. So I'm going to tell you and then provide my reasoning. So CTF decreases the number of crossing over events. What would that have to do with the number of copies that are distributed during meiosis? So this takes us back to here, which says crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the first cell division. So we've established that CTF decreases crossing over. Now, if crossing over is required for proper division, decreasing crossing over should decrease proper division, and it should decrease the accuracy of the divisions. Okay, so for this one, I would say, so predict the effect. In terms of the number of chromosomes, overall, I'm pretty sure there could be a couple different answers for these, because overall you would just get non-disjunction. So you could say either that some cells would have more or less. So +1, -1, overall they would be a non-disjunction event. So you can't say for certain. They probably would have accepted both, or you might have need to say both, but non-disjunction would occur. So you'd say non-disjunction, where some have extra and some are missing. And the reason for that, so my justification is that CTF decreases crossing over. And then since it's required, for proper distribution of chromosomes, you're going to get non-disjunction, or basically less accurate moving around of those chromosomes. So this is the bulk of the question, that you have some with extra and some with missing chromosomes. And then again, the reason for that is since CTF decreases crossing over, it should also decrease the accuracy, or how well the chromosomes are able to separate, and that's going to give you your non-disjunction. Okay, now for Part 3, it says, explain how the presence of hotspots seen in Figure 1 could increase the likelihood that a population will survive in the presence of selective pressures. So this one actually doesn't have to do too much with the question. We need to know the purpose of crossing over. Now, we know the purpose of crossing over is going to be for genetic variation, and that's actually also the reason that certain populations survive in the presence of selective pressures. So we know, and I think we'll come to a question that also addresses this, that low biodiversity or low genetic diversity is not great for populations or ecosystems, because if there is some sort of pressure, or there is some kind of disturbance, they're going to be very hard to survive and bounce back from. So what you want is genetic variation, and that's what crossing over's purpose is. So the presence of hotspots would be good, because I'll just pop this up here. So increased crossing over or hotspots increases genetic variation, which should result in higher resilience. Again, you didn't need to put this exact wording, but the general gist of the fact that crossing over is going to increase genetic variation, and we know genetic variation is going to help populations survive under some sort of pressure. Okay, so this was number 1. It wasn't too bad. Here are all your answers for them. This one was probably worth about 10 marks. Okay, so this is number 1.